Module 6, Objective 1, and I noticed the typo here. Let me just go ahead and correct that very quickly. So this should be Module 6, Objective 1. Asks that we identify the major regions of the brain, and then it lists the 6 major regions of the brain. The brain itself is rather complex, and there are 6 major regions that are all color coordinated here on this figure. For the most part, I think the general population would identify this as being the brain. This is a very large, important region of the brain called the cerebrum, but it's only one part. In addition to the large lobes of the cerebrum and the cerebellum here in blue, there's also some internal structures and some basal structures that are collectively known as the brain stem. And that's what we have here, the brain stem. And we see that the brain stem is comprised of many parts. If you follow up from the spinal cord, we have the medulla oblongata, the pons, you have the mesencephalon or the midbrain in green, and then lastly the diencephalon, which comprises, it's made of both the thalamus and the hypothalamus. Overall, the brain is a very large, delicate mass of neural tissue. It contains a number of internal passageways and chambers that is filled with fluid, namely cerebrospinal fluid. There are 6 major brain regions, each region having specific functions. And as we ascend from the medulla oblongata, from the spinal cord through the medulla up to the cerebrum, we find that the brain functions become more complex. In fact, often our humanness is attributed to the functions of the cerebrum, which covers higher level thought processing and the ability to critically think. Conscious thought and as well as intelligence are also seated within the cerebrum, or within the cerebral hemispheres of the cerebrum. Objective 2, explain how cerebral spinal fluid, meninges, and the blood-brain barrier support and protect the brain. There are several ways in which the brain is supported or protected. The physically protected from the bones of the cranium. We know that the cranium surrounds and protects the neural tissue. In addition to that, there are 3 distinct layers of cranial meninges. Meninges are layers of connective tissue that sort of surround the central nervous system, both the brain and spinal cord, almost like bubble wrap. So we have 3 distinct layers of this connective tissue, and that really protects the brain from the skull. If the head was to be hit, you can imagine that delicate neural tissue hitting the bone of the skull and causing damage. So by covering the neural tissue with these cranial meninges, it sort of bubble wraps that neural tissue and imbibes a bit of protection. Lastly, we have the actual fluid, the cerebrospinal fluid. This is found between the layers of the meninges. If you use the bubble wrap example, we know that sheets of plastic are separated by air pockets. In the case of the meninges, you have fluid pockets, and the fluid is the cerebrospinal fluid. But CSF also flows within the passageways and the open cavities within the brain. And we'll cover the anatomy of all these structures in lab. But for lecture, you just have to know that that's where we find the cerebrospinal fluid. In addition to the physical protection, we also have biological isolation. For the most part, blood and brain do not come into contact with one another. And the main reason is because blood fluctuates too frequently. If you consider perhaps blood sugar levels, after a meal, it's not uncommon to have blood sugar levels shooting up hundreds of points. So the brain really can't handle that instability. So there is a blood-brain barrier that keeps the two separate. In addition to just sugar levels, it's the levels of hormones and other chemicals that we find in our blood that the brain can't handle in terms of the fluctuation of concentrations. So the blood-brain barrier really keeps those separate. We'll start with the meninges. Again, there are 3 layers, and this image is really good if you take the time to look at it. First, note the placement of the cranium. So this is the bone of the cranium. The first layer is the dura mater, followed by the pia mater. Well, I'm sorry, I said that backwards. The dura mater is followed by the arachnoid mater over here. And then most deep is the pia mater. So from superficial to deep, you have the dura mater, the arachnoid mater, and then the pia mater. And in between each of these layers of meninges, there are often spaces. So you're going to find things like the subarachnoid space, situated between the pia mater and the arachnoid mater. Or you might find the subdural space. If it's present, the subdural space is situated between the dura mater and the arachnoid mater. The dura mater consists of 2 fibrous layers, an inner and outer. The outer fibrous layer is known as the cranial dura, or the periosteal cranial dura, because it's continuous with the periosteum of the overlying bone. The inner fibrous layer is known as the meningeal cranial dura. The dura mater follows crevices within the brain. If you consider an anterior view of the brain, and my drawing skill is not very good, but you have the left and right cerebral hemispheres. And in between the left and right cerebral hemispheres, there is a large crevice right here. And what we find is that the meninges, specifically the dura mater, it actually follows down within that crevice. And we can see that right here is at the falx cerebri. This layer of tissue follows down into that longitudinal fissure that separates the left and right cranial hemispheres, or cerebral hemispheres. There are 2 other folds that are noted down here, the tentorium cerebelli and the falx cerebelli. And the role of these is to stabilize and support the brain. But it also allows for the collecting veins to be distributed or carried deep within the neural tissue. So we actually have veins that are traveling, and we can see them here, these large blue vessels. Right here is a few. They're going to follow these dural folds down into the deeper recesses of the brain so that they can do their job. So I already noted the falx cerebelli and its placement between the cerebral hemispheres. The tentorium cerebelli separates the cerebrum from the cerebellum. And if we go back one slide, we can actually see that right here. So you would anticipate the cerebellum to be occupying this occipital lobe, or rather the occipital bone, and it's going to separate the cerebrum from the cerebellum. And then the falx cerebri is right here, falx cerebelli, and it separates the left and right cerebellar hemispheres that are most posterior. So that was the dura mater. The arachnoid mater is the middle layer, and it creates a space called the subarachnoid space. And I hate to flip back and forth, but it's probably worth doing. If you look here at the subarachnoid space, right here, you can see that there are lots of little fibers that reminded early anatomists of spider webs. So the arachnoid part of the name actually stem from the appearance of these fibers. But it creates that subarachnoid space. And then lastly, we have the pia mater. It is the deepest, most layer. It's actually attached to the surface of the brain via astrocytes. So it's continuous with the surface of the brain. Cerebrospinal fluid. It functions in support. It helps to cushion and absorb shock. And it also transports nutrients. Since there is a blood-brain barrier, you can't count on blood to distribute nutrients. So the nutrients are passed on within the cerebrospinal fluid, and the cerebrospinal fluid then distributes nutrients as well as carries waste products away from neural tissue. Within certain parts of the brain, we have collections of ependymal cells. Remember, ependymal cells are a type of neuroglial cell, and the role of that cell is to produce cerebrospinal fluid. So when you have collections of these ependymal cells that surround capillaries, here's our capillary, you get formation of the choroid plexus. So these are specialized ependymal cells that surround capillaries, and they're going to secrete about 500 milliliters of cerebrospinal fluid into the ventricles per day. And I know, again, we haven't covered ventricles yet. They're going to come up in lab very shortly, but for now, we'll leave that term there. So as 500 milliliters of CSF are being produced each day, it also has to be drained each day. So we have to have a way to drain that. And the role of CFS is to really, again, distribute those nutrients and carry the waste products away. So the choroid plexus is going to adjust the composition of CSF to provide the adequate nutrients. It also carries away any waste products that is collected within the cerebrospinal fluid. In terms of circulation, it's going to circulate from the choroid plexus through these ventricles, these large cavities within the brain, down through the central canal of the spinal cord, which we've already discussed, and then up through those subarachnoid spaces. So it flows both internally inside of the brain, cavities and passageways, but also through the meningeal layers on the outside. In terms of draining, when the cerebrospinal fluid is in the subarachnoid space, as we see right here, it drains through the arachnoid villi. Villi are nipple-like projections. So you have these little interruptions within the subarachnoid space that are the arachnoid villi, and they extend directly into a vein. So here we have a vein. It's not labeled as a vein, it's probably labeled as a sinus, but yeah, right here, superior sagittal sinus. But it's in all respects, it's a type of vein. So it's going to be mixed back into the blood, where then it's going to be drained from the head and then put back into circulation. Collections of the arachnoid villi form arachnoid granulation, and that's pretty much what we have here, these large regions where the cerebrospinal fluid is absorbed back into circulation. We noted the presence of the blood-brain barrier, or at least we talked about the reasons why we have to have the blood-brain barrier. In terms of the BBB, we see that it isolates the central nervous system from the circulation. Here we're just talking about blood. It's formed in a number of ways. One, we have tight junctions. Remember, tight junctions are cellular junctions that latch epithelial cells to their neighbors, and in doing so, they isolate the lumen. So that's going to help within the endothelial or the epithelial layer of the capillary by creating a strong bond that prevents blood from encountering neural tissue. You also have help from the astrocytes. Astrocytes, if you recall, are neuroglial cells. They also surround capillaries and help prevent the movement of blood into the neural tissues. We saw an image of an astrocyte, and I'm going to go back a few slides to point that out. Here it is right here. These little yellow cells in this figure. You can see how these yellow cells are sort of surrounding this capillary, and in doing so, they're really going to create a barrier to prevent the mixing of blood with neural tissue. So they do a very good job of helping to maintain this blood-brain barrier. There are 4 breaks in the blood-brain barrier. 3 of them are hormonally, are due to hormones. So portions of the hypothalamus, the pituitary gland, and the pineal gland. These are hormone-producing glands or parts of the brain that that exhibit a break in the blood-brain barrier, mainly because hormones have to be transported by blood. So you have to have a way to get the hormone to the blood. So this break allows for that. The other break that we find is at the level of the choroid plexus. And again, since it's the role of the ependymal cells of the choroid plexus to create cerebrospinal fluid, you have to have blood as a precursor, because blood gives rise to cerebrospinal fluid. Or it may be better to say that cerebrospinal fluid stems from blood. So a break there makes sense as well.